I'm also from Madison, Wisconsin. I'm at the USDA ARS, so I work in Phil's group, but I'm also in entomology at the Department of Entomology at the university. And Megan Van Hetten was a postdoc of mine. She was at Massey University, and now she's back in the States. But we started this study together when she was in my lab. So when I'm trying to do this approach, for many of you or most of you today will probably be quite different. So you have to think about wild population. I'm an evolutionary ecologist by training, so I think about wild population and the diversity of wild population. I want to introduce an approach that can be used to look and think about carrots and why carrots are invasive. They are very invasive in the States. I've been told that I'll see them everywhere here too in France when I drive around. So there's ways in ecology where we can study a population and try to determine what life history stage contributes to the invasiveness and population growth of a population. So this is what I want to introduce to you today, how we do this and how helpful it can be. So you can also use just the ecology, and you can, at the end, we're starting to look at how you can combine the population dynamics and population ecology approach with population genetics. So you can look at specific genes and look at the spread of those genes in a population. So relative to what we've talked today, in some ways, any genes that came from cultivars to wild species, you could try to see what will happen to the genes in the wild species. Similarly, if you could find genes that are specific to Gummifer, then you could look for that gene and try to study its spread in the wild population of Carota carota. Okay, similar things. So what I'm going to do today is first I'll introduce a study we've calculated empirically different life history stages: reproduction, germination, survival, and flowering. And these are the kind of data that are helpful to put in a population matrix model, so you can put the basic information you get from a population in a model to predict whether population will grow in size and see what will happen to that population size over time. You can also look using what we call sensitivity analyses, what stage of the life cycle is most sensitive to population growth. So if you change, for example, germination, if you increase it, will it have a bigger impact on population growth than if you change reproduction? So you can establish which part of the life cycle most affect population growth. And if you're interested in control, you can concentrate your control efforts into this stage of the life cycle of carrot. So, and at the end, I will show we're just starting to combine population dynamics with population genetics to look at single genes and how they would move in wild carrot population. So this study was done in Madison. We look at 2 wild populations of carrot in Madison. And let's, for simplicity, think that it's most of the carrot individuals there are, let's say, biennial individuals. So in Madison, that would mean producing seeds in the late summer and fall, germinating the following spring, growing in that summer, surviving as rosette over the winter, and flowering the second year, if the first year of production of seed is year 0. In contrast, if you had a winter annual, because we have strong winters, unlike you guys here, we produce seed in the summer, they would germinate in the fall, overwinter as rosette, and they would flower and die in the spring. This plant is monocarpic. Once it's flower, it dies. So this is just to show you what the Madison population looks like. It's like probably where you find it anywhere or most places along the roads. It's everywhere in Madison in late July and August. This is what we call the Middleton population. This one is a little different. It was a restored prairies, but all the carrots invaded. So all the carrots you find in that population come actually from the other wild population around it. So the stages of the life history we concentrated on was reproduction, seed set, seed germination, rosette survival, and the flowering rate of these populations. So the basic model we want to feed our data in is the following. You imagine a reproductive, and the reproductive would set seed, and we call that year 0. Then those seeds would germinate and the following spring, and then they would live as rosette in year 1, and then they would survive the winter. So we looked at from primary to quinary umbel types to determine the reproduction of our plants. So what we did to look at reproduction, we set up plots in a population, 3 by 3 meter plots. We marked 20 reproductive per plot, so we had 200 reproductives in Middleton and Madison population. I prepared this while I was jet lagged. So what says Middleton is actually Madison, and what says Madison in that slide is actually Middleton. So in 2010, we looked at the Madison population, and in 2011, we looked at both Madison and Middleton population. Recorded the number of umbels at each level on each plant, so we have 200 plants per population each year. In order to get the seed set from this, because these plants produce a huge number of seeds, we decided to select 50 individuals that were flowering. And on each individual of these 50 individuals, we bagged at most 3 umbel per umbel type. So you can't have usually 3 primary umbels, so we bagged whatever was a primary umbel, and then 3 secondary, 3 tertiary, and same with quinary, you don't have 3, so we bag whatever was there. So we bagged it before the seed production, so we wouldn't lose any seeds. Then we removed the seeds and we weighed them. So we had to go through the whole umbel and removed all the seeds. We weighed the total seeds per umbel type that we have for each umbel. Then in order to really get a seed count, because counting those seeds would be a tremendous job, you can do, we weighed 3 sets of 10 seeds, took the average, and used that information to get the total seed numbers from the total seed weight we had for that umbel. So then we determined the seed number per umbel level in that population, or for each plant. And then to determine the total seed count on that plant, we knew how many umbel at a certain level we had, and we multiplied that by the number of seeds for that umbel level for that plant. And we added that up for every type of umbel, and that gave us a total number of seeds per plant. Okay? So when we did that, we found these are the numbers. So in the population on average, what we found, this is average over year and the 2 population. There was overall about 1 primary umbel was typical in the wild population. We had about 5.6 secondary umbel, 9.4 tertiary. So there were more tertiary umbel than secondary umbel. Very few quinary umbels. But if you look at seed set, the primary umbel set way more seeds than all the other umbels. And because the tertiary umbel set way fewer seeds than the secondary umbel, overall, when you look at the contribution of the umbel type to reproduction, what you find is most of the seeds on a plant actually comes from a secondary umbel. And the first and the third provide about 20% of the seed set of the plant. So that's what happened in our population. What we found was that each plant on average, so this is average over the 2 years and the 2 population, produce about 4,400 seeds. So these plants produce a lot of seeds. We found no differences, though, among the populations. So the 2 years, we could compare the population in 2011, and we could compare the Madison population between the 2 years. So we found no differences either between the 2 years in the number of seeds set per plant. What we did to look at the germination, we took a primary umbel on 80 individuals from the Madison population in the first year, and then 20 individuals from the other 2 populations in the second year. From each of these umbel, we grew 50 seeds, so in a single pot. Okay, so what we found was rosette survival over the winter was 49%, and the flowering rates of those rosette, the one that flowered, was 6.3%. So where we have now, to just summarize the empirical data we gathered from these populations, the seed set was 4,400 seeds. There were two germination rate. One was high, 30.9%, and the low one was 2.7% of our seeds are germinated. Rosette survival was 40.9%, and flowering rate was 6.3%. So to look, we can now use these data. This is a very basic, and you can elaborate much more complicatedly. But the basic population model, we would have a reproductive stage. The reproductive stage would become our non-reproductive. And the transition stage for between reproductive and non-reproductive would be the number of seeds that were produced and the percentage of those seeds that germinated. Then the non-reproductive would have to overwinter, and they'd flower. So the ones that flower would die. Okay. And the other ones would remain as rosettes. So this loop here is the ones that are not flowering and remaining in the rosette for the next year. So this basic model, you can run it over time to see what would happen to population growth. What we find is that with the low germination rate of 2.7%, the lambda was 1.95. That means any lambda greater than 1 indicates that your population is growing in size. So even with a 3.9 germination rate, your population is growing in size. With a high germination, the lambda is 6, so this population is growing even more in size. In case I don't make it, after 3 years with a low germination, you'll have about 383 individuals if nothing changes. With the high germination, you will have 47,000 plants in your population. Okay, if you leave one to reproduce. Okay. What you also see is most of the individuals in the population are predicted to remain as rosette, and a small percentage of them are reproductive. The sensitivity analysis tells you if I change any stages of my life cycle, so any of these transition. So by this transition, we mean here the seed time germination, for example. I don't know how to work this. So if you change either this one or this one, how will that affect population growth? And this is what the sensitivity analysis is doing. So it's telling you here that the most sensitive period or transition is the non-reproductive to the reproductive. So the one of overwinter survival and flowering rates. So this is way more, if you control a population, controlling at that stage will give you way better result than if you try to simply reduce the seed set or lower the germination rate. So this has very practical application that have, I'm sure, I mean, I know that the way they control the carrots is, at least in our part of the world, you mow and you kind of put herbicides on the rosettes. Okay, so I want to make sure that you understand when you mow, please mow before the plant flower, and please do not let any plant flower, because even if you really want control, leaving one plant, if your germination is 30%, after 3 years, you predict that population will have 47,000 plants. Okay. So in summary, under both kind of model, you have population growth. This plant is invasive. This is why, okay? This is a number, this is modeling that helps you see why population are invasive. And what is it about the stage of life of the carrot that you can control, use most to control. And you can do these studies in more details to really go back to more detail about which stage it might be. Okay. So I've said all this. So what I want to tell you is now we've also started looking about, you can have annual and biannual in the population, and you can look about, will the annual be better than the biannual? Will they grow faster? Under what condition would an annual grow faster than a biannual? You can also look at hybrids, and we know because some people have done studies of looking at the life history, the reproduction and survival of hybrids. You can plug data on hybrids in these type of model and determine what will happen to your hybrid in the population. And we've done some simulation looking at this. The latest we're trying to do is to combine population genetic and population dynamics. And just to give you an idea, this is kind of thing you can look at, okay? So you can look at the genes by themselves, okay? So you can kind of separate the genes from the presence of an hybrid. And most of the population simulation we're running is telling us that a genes like Roundup Ready, okay, which is neutral in a natural population, so it's considered a neutral genes. Well, these genes will, in many cases, remain in a wild population. Okay, not always, and it will depend on population size and things like this, but in many cases, if you run enough cases, some cases it will remain, and once it remains, you can see it spread through the population. So these kinds of models can be done, this is done within a single population, but if you include gene flow, you can also see the spread of your population. So it could be useful when you're thinking about genes that flow from cultivated to wild and try to determine what might happen to these genes in a population. So anyway, this is just another simulation we've been doing, looking at the same thing, that over 100 times, there will be times where those genes will be maintained in a population, even if the hybrids are no longer there. Okay, so I will stop here. Oh, and I want to thank, I prepared this, so I want to thank the USDA ORS for actually supporting this research, and I'll take any questions. Thank you very much.
Thank you very much for your very practically made research. Very interesting. But it's, I wonder, maybe it's possible. If not, did you see did you look at the size of the seed of carrot, for example? Because it's sometimes the seeds in carrot you can find out the seeds can be small, but the germinations can be good and otherwise. Yeah. Maybe size of the seeds can be the key, maybe the trait you could look after. Thank you.
My answer to this would be, in a tough year, you had 2.7% germination. So if you look, it could very well be that those were the largest seed in the population. We don't know. But, that could be very well be what it is. That in a good year, it doesn't matter your size. It could be that in a bad year, your size will really matter. And then you make the population even stronger, because if that's the case and you keep reproducing, and if there's any heritability to seed size, after a few generations, the large seeds will make it through natural population, and then it will make it, their germination could even go up. So, yeah.
Any other questions?
I don't know if you have some information about to which stages of the vegetation evolution can your studies better refer? Because, of course, carrot is an invader at some stage of the evolution of vegetation from a crop, a previous crop, then through its evolution. So your applied result, to which kind of management you apply, because if you go to really cultivated fields, still cultivated, you find no carrot. Then in an old field, a few years, abandoned from a few years, you find carrots, can be the case of this orchard, biological orchard, but then at further stage of vegetation, carrots tend to disappear again. So...
because there's competition. Okay, so you could, So this is as if, in these models, these are population dynamics model, you can put carrying capacities. So these basic model I presented don't have carrying capacities. So you can decide what's the carrying capacity. What you're referring to is with succession in natural environment, then competitive ability becomes important. And carrots may not be very competitive compared to other species, so other species will take over. So you could build that in by changing the carrying capacity of your, you decided over time there'll be, so you change. This is more of an exponential type of growth at this stage. This one has no competition. Yeah, it's the ideal world for carrots. But it's just to give you an idea of how it works. So we could put it. I just couldn't present everything, but you can do that. Yeah.
One last question, Bodo.
I mean, just first to comment and then my question. It's quite astonishing with this survival rate and with this reproductive rate why our meadows are not completely full of carrot plants all the time. Okay.
But they can't always get there.
Yes, they can always get there. Yeah.
Thanks God.
What I would like to know is whether you have any experiences or experiments on the survival rate of the seed that did not germinate in the soil. How long does
seed
it take?
bank. Mm-hmm.
And the survival rate and the fate of these plants that remain in rosette state to the following years to come.
Well, what I would like is, I don't know much about seed banks. What you're referring to is a lot of wild plants. They remain alive for a number of years in the seed bank, and you can put that in. We don't have data on that. The other thing that would be really nice, that right now we've made it really simple between reproductive and non-reproductive. I would like to have different sizes of rosettes, because I have a feeling a lot of these plants are actually not biennial. Some are annual, we know that, so we've played with that, but I have a feeling some are triennial and, I don't know how you'd say 4 years, but they've become somewhat perennial. So if we really had the size of the rosettes and data that I'm presenting on each size classes, then we could really get a better feel for, the real life cycle of wild carrots, which I think is way more complicated than just a biennial plant.